Thanks, Angie. Good morning, everybody. I'm not sure this was really the best time to plan a talk on neurobiology. I feel particularly bad for the people coming from the West Coast who are probably struggling to stay awake. And so I'm going to try to provide a pretty simple and straightforward overview of the circuits that control sleep and wakefulness. And the idea is to sort of set a foundation so that we have some anchor points to understand the importance of the various neurotransmitters and so forth that are good, pharmacologic targets. And so I'm going to do a very quick and sort of high-level review of what we understand about the neural circuitry that controls sleep and wakefulness. An even shorter overview of the circadian circuitry, and then try to sort of tie it together with how this is relevant to drug development and what are some of the targetable sites within these various networks. I don't really think I need to tell this audience what sleep's all about, but of course we distinguish these states oftentimes by the electroencephalogram, low amplitude fast activity in wake, varying degrees of slow wave activity or delta activity in non-REM, and then REM sleep is sort of a wake-like state, but of course it has very important distinctions. And one of the things I just want to emphasize is just how sleep changes across life. And we are often developing drugs to target specific diseases, but certainly anybody who has kids understands how kids sleep differently than adults, and young adults sleep differently than older adults. And so here's a couple hypnograms from a young adult, 23, and an older person, 68. And you can see the typical sleep cycle that's occurring across the night with alternating periods of non-REM and REM sleep, the REM sleep shown in the black bars there. And so the 23-year-old falls asleep quickly around midnight, goes into some deep non-REM N3, and then eventually is waking up around 7:30 or so. The 68-year-old has a very different pattern to their sleep. They show the same alternation of non-REM and REM sleep, but the sleep is much more fragmented. They're spending no time in deeper stages of sleep. In fact, it's not at all unusual to see N3 completely disappear, once people get into their 50s and 60s. And then there's some more tendency towards an earlier awakening. And so it's important when we're thinking about drugs to think about who we're treating as well, because this tendency towards lighter sleep and early morning awakenings and this fragmentation are all things to factor in. So turning to circuitry, when I was in medical school, and I think still at many medical schools, people refer to the reticular formation as being the source of the arousal drive. And we were taught that if a patient has a lesion at the top of the midbrain, that is a common cause of coma. And anatomically, it's a fine way of thinking about things. But over time, we've really come to appreciate that this idea of the reticular formation is just really not correct. There are these neurons that you could call the reticular formation that run the length of the brainstem, as shown here, this little sort of purplish color running from the medulla up to the midbrain. But it's not a monolithic entity. In fact, there's a lot of different cell types that send their projections through these regions. And so over the years, we've come to identify these. It's really been an effort, really since the 1980s or so, to tease apart what are the specific types of neurons, what do they connect to. And some of the new techniques that I'm going to just touch on very briefly are really now allowing us to dissect these circuits in much greater detail. And so this is a fairly dense summary of what we think are some of the key wake-promoting pathways in the brain. And we've broken them down here into two different categories. There's the monoaminergic systems, making monoamine neurotransmitters, and those are shown in the light green. And those include some areas that are familiar, such as the dorsal and the median raphe nuclei that make serotonin, 5-HT, dopamine cells in the ventral tegmental area and possibly some other brain regions, histamine neurons in the tuberomammillary nucleus, and noradrenergic neurons, abbreviated NE, norepinephrine in the locus coeruleus. And the monoamine neurons, in general, project diffusely throughout the forebrain. They have typically excitatory effects. These cells are active during wakefulness, minimally active in non-REM sleep, and they fall silent in REM sleep. And that loss of activity in REM sleep is actually very important, as we'll come back to later. And they're probably all active at different times in different circumstances. And I'll try to tie this together a little bit as we go along, but it's just important to remind you all that when we talk about wake-promoting pathways, yes, all of these pathways help promote arousal, but they're probably all doing it in their own special way. And so, for instance, we know the locus coeruleus neurons particularly fire when an animal is engaged in recognizing salient stimuli in the environment. When you really have to focus and pay attention on something, that's what your norepinephrine system is about. The dopamine neurons in the ventral tegmental area are certainly turned on when an animal is engaged in going after some reward, and there's something they're very motivated to accomplish, and you need a high level of arousal to go after that target. And what, for instance, engages the histamine neurons is still something of a mystery, but they're certainly important. So those are the monoamines. And then there's other groups that we could just sort of loosely lump as the cholinergic cell groups. And the basal forebrain is probably the one that you're most familiar with. These are the cells that degenerate in Alzheimer's disease. And these neurons predominantly project to the cortex. And the acetylcholine neurons in the basal forebrain directly excite cortical neurons. But there's also GABA neurons in the basal forebrain. And you say, wait a minute, GABA is an inhibitory neurotransmitter. Wouldn't that be inhibiting cells? Well, what it does is it actually inhibits inhibitory neurons in the cortex, thereby disinhibiting other cortical neurons. And so the net effect is to activate the cortex. There's also sort of poorly understood glutamate cells in the basal forebrain. But pretty much you can think of the basal forebrain neurons as directly activating the cortex, whereas those monoamines are activating neurons at all different levels of architecture in the central nervous system. In the pons and low parts of the midbrain, there's also another cholinergic area called the pedunculopontine and lateral dorsal tegmental nucleus. Probably like worst name ever in the brain. And these cells don't project to the cortex. These guys activate subcortical regions. In particular, they help turn on the thalamus. And so when they release acetylcholine into the thalamus, it turns the thalamus into a state where it can actually relay information to and from the cortex. If you don't have acetylcholine in the thalamus, the person's not going to wake up. And so it's necessary for flipping the thalamus into that state so you can have full consciousness. There's also cells in that area that make glutamate and GABA, and we're trying to figure out what those do right now. There's another region, I've colored it the same, but it's not actually cholinergic. It's that dark green called the parabrachial nucleus. These are glutamate neurons. And these are just, their role in sleep was really just recognized in the last few years. These cells are neat. These cells are activated by noxious visceral stimuli, pain, cold, and interestingly, hypercarbia. And so just in the last few years, Cliff Saper and his group have identified that these cells are crucial for waking people from sleep when there's airway obstruction and the person becomes hypercarbic. And so if that area is turned off or lesioned in animals, and then the CO2 level rises, it takes them much longer to wake up. And so those parabrachial nuclei neurons can be considered an important sensory relay that helps drive arousal in the forebrain in response to things like hypercarbia. It's fun because it's actually sort of the beginnings of a targetable system for pharmacologically addressing obstructive sleep apnea. So just to give you a taste of how people are now teasing apart these pathways, this is some work done by Louise Delathea's lab, by Matt Carter, and this is using optogenetics. And optogenetics is a technique where we can put these rhodopsin-like molecules into neurons, and then we can turn the neurons on by shining laser light, or you can put a different type of opsin in, and you can turn the neurons off. And so on the left-hand side, we're using Matt was using an excitatory channel rhodopsin to turn on locus coeruleus neurons in different states. And what you can see there is that if you activate these neurons at about 10 hertz, the animals have about twice as much wakefulness as in the baseline condition. Putting the other kind of opsin in that's inhibitory, if you inhibit those locus coeruleus neurons, you see a small reduction in wakefulness. That's shown in the yellow and black bars over there. But more importantly, if you shift to the right panel, where you're looking at transitions between states, what you can see is that activating the locus coeruleus causes considerably more transitions from wakefulness to non-REM sleep, and so sleep becomes very fragmented. And so these neurons are important for helping wake you up. And the real beauty of optogenetics is it allows you to manipulate these neurons on a very fine time scale, and we can do it in a very cell-specific and anatomically specific fashion, and that's really helped us be able to break apart these circuits in much greater resolution than some years ago. I briefly mentioned histamine before, and histamine is one of the more interesting neurotransmitter systems. We think it's just as important as any of the other monoamines, but we just know a lot less about it, and really we need to do much more research. The histamine neurons project diffusely to the whole forebrain. They help activate the cortex and other regions, and we know that animals that lack histamine, like if you acutely block the histamine system, that's sedating. But nobody's ever really quite tied it into specific diseases. And then some years ago, Seiji Nishino and Kanbayashi measured CSF histamine levels in patients with a variety of different hypersomnias. And what they found was that patients who had narcolepsy with cataplexy tended to have low histamine levels in their spinal fluid. Same thing for narcolepsy without cataplexy and idiopathic hypersomnia. But patients with sleep apnea, who also have sleepiness, of course, the range of values was not terribly different than that of the controls. And so this raised the possibility that maybe low histamine is a common theme in some of these central nervous system hypersomnias. I have to say, when this first came up, we were all really excited about it because it was providing some of the first clues as to what might be going on in particularly idiopathic hypersomnia. But there's been some subsequent studies by Dovier and others which have said, well, maybe the histamine levels aren't all that different. And honestly, it's a very mixed bag right now. Half of the papers that have measured histamine say that it can be low. The other half say it's normal. And it's sort of there's a big fight about what's the right technique for measuring this. But I wanted to sort of throw it out there as a possibility that histamine may have a direct relation to some of these disorders. Dopamine is really worth spending a little time on because the dopamine system is pretty much the most common target of wake-promoting drugs that we use in clinic. And we know that if we give drugs that increase dopamine tone, that wakes you up. Dopamine levels are generally high during wakefulness spontaneously, and amphetamines predominantly work by increasing dopamine levels in the brain. They block the dopamine reuptake transporter so that when dopamine gets released, it doesn't get taken back up into the presynaptic terminals as quickly, and you have high dopamine levels at the synapse. Modafinil probably does something similar, but in a more specific way. Mice that lack the two main types of dopamine receptors, the D1 and the D2 receptor, don't wake up with amphetamines, or the response is substantially blunted, showing that that dopaminergic effect is pretty crucial. And mice and flies that lack the dopamine reuptake transporter also don't wake up with amphetamines, and they don't wake up with modafinil either. And so that's pretty strong evidence that high dopamine helps promote wake. And the flip side is also true, that if dopamine tone is low, you're going to be sleepy. And certainly in the hospital, this is used quite commonly when you need to help get patients to sleep. They're giving antipsychotic, anti-dopamine drugs quite frequently, like haloperidol. In Parkinsonism, of different cause, people are often sleepy. In part, that may be due to this dopamine deficiency, but there's lots of other reasons why Parkinson patients can be sleepy. And some drugs that we give clinically, like pramipexole and ropinirole, are sedating, and you're like, wait a minute, these are dopamine agonists. But what's happening with those drugs is they're acting upon the D2 receptor, which is an autoinhibitory receptor on the dopamine neurons, so that we think that when we give pramipexole, it's actually reducing activity in the dopamine system, and so the net effect is to actually have less dopamine tone, and that's where the sleepiness with those medicines can come from. So it's a bit of a double-edged sword, because we use these in patients with restless legs with the goal of helping them get more comfortable at night, but then the drugs can be sedating, and that daytime sleepiness can become a problem. I mentioned the basal forebrain. I don't think I actually need to say much more on it because we covered this already. The cholinergic neurons excite the pyramidal cells. People have now used optogenetics to selectively turn on those cholinergic neurons. And if you do that when an animal's in non-REM sleep, they can't produce slow waves anymore, because if you activate the pyramidal cells in the cortex, it's running counter to the rest of the brain trying to produce these slow waves. If you reduce activity or lesion the cholinergic neurons, the cortex can't produce the typical fast frequencies of wakefulness. And so the cholinergic neurons are very important for those two things. They help drive fast cortical activity, and they help suppress slow waves. The GABA neurons are mainly involved in promoting wakefulness. The glutamate neurons are pretty mysterious still. If you activate them, they can promote wake, but we don't really know how that occurs just yet. There's a lot of papers written on basal forebrain lesions, and many of them don't show a whole lot of effect. And part of the problem is the basal forebrain is actually kind of large. It runs sort of from the front end of the hypothalamus right up under the septum. And so a lot of those papers doing lesions didn't see much, but if you really thoroughly kill off the whole basal forebrain, animals are basically comatose. And so I think you can really look at the basal forebrain as one of the final common pathways to the cortex. And if you don't have a basal forebrain, you really cannot produce normal wakefulness. So I just want to come back and just sort of reinforce this theme of, when we talk about wake-promoting systems, that they probably each have their own flavor. And so, different parts of the cortex receive varying amounts of these inputs. And so, we think that attention and alertness is very dependent upon norepinephrine, histamine, and acetylcholine. But if you're talking about waking up for the purpose of going after some rewarding goal, whether it's food or drugs or whatever it might be, then dopamine and acetylcholine are important. And of course, if you're talking about arousal in terms of emotions and anxiety, serotonin, dopamine, norepinephrine, and glutamate are going to play into that. And then you can think about arousal from an autonomic perspective, where to some degree it's actually descending projections that are most relevant. And there, norepinephrine and other things are probably playing into it. So just, we tend to refer to wake-promoting systems and wake-promoting drugs, but I just really want to reinforce the idea that it's good to think about the specific type of drug that you're considering, because it's going to have different effects on the kind of wakefulness that will result. So switching gears to non-REM sleep, I don't think I need to remind you all what this is about, but basically, you see varying degrees of cortical synchrony and high amplitude activity. When somebody's in very deep non-REM sleep, N3, it's hard to wake up. They have sleep inertia. However, oftentimes people do have some sort of mentations that, in deep non-REM sleep, they're often fully unconscious, not thinking of anything. But there is sometimes some dreaming, but the dreams aren't vivid like they are in REM sleep. And parasympathetic tone tends to be on the higher side. So heart rate is slow, breathing is slow. And until about, late 1960s, 70s, people thought that non-REM sleep was just this passive thing. The arousal regions just somehow turned off. They got tired in some way and turned off, and that's how sleep occurred. But it became clear that there's cells in the preoptic area that are actively involved in driving sleep. And one of the cell groups there, the ventrolateral preoptic area, has been studied quite a bit. It's a GABAergic cell group, and it innervates all of those arousal regions we just talked about. And so if you record from cells throughout the brain, like Ron Samusiak has done, most times you're going to find lots of wake-active cells. There's very few in the brain that actually fire more in non-REM sleep, but these cells in the preoptic area are one of those few populations. And so what's cool is they're inhibitory, and they turn on somehow during non-REM sleep, and they turn off these other wake-promoting regions. And so we really think of them as being involved in helping coordinate the inhibition of the arousal systems. And there's a few different preoptic cell. So the preoptic area is the front third of the hypothalamus, and there's a few different cell groups there. The median preoptic area has gotten some attention too. There's also a recently discovered group, and I only mention it for completeness. It's called the parafacial zone. It's down in the lower part of the pons there. And this does not globally shut down the arousal regions. It seems to predominantly innervate the parabrachial nucleus, that area that was involved in responding to visceral stimuli. And its broader role in sleep is something that's being sorted out right now. So just to give you an example of how important these preoptic cell groups are, this is 24-hour sleep recordings from rats that before there was a lesion and after there was a lesion in the preoptic area. And in this particular case, the lesion's focused on this ventrolateral cell group. And so in green, what you can see is a strong diurnal variation in non-REM sleep across 24 hours, and rats, of course, are active during the night period. And so what you see is that during the dark period, shown in the black bar at the bottom, non-REM sleep is occurring only maybe 30–40% of the time, and during the light period, the rats are awake about 80% of the time. Sorry, they're in non-REM sleep about 80% of the time. But after the ventrolateral preoptic lesion, their non-REM sleep is now reduced to about half of what it was at baseline. And so even during their main sleep period, the light period, they're only having maybe 40–50% time spent in non-REM sleep. And what's remarkable about this is often with brain lesions, we see these transient effects where animals are, less awake or more awake for, a few weeks. But this persists for months. Jun Liu recorded from these rats 3 months after the lesions, and they still hadn't recovered. And what it really tells you is that there's nothing else in the brain that can pick up the slack. The preoptic area is really crucial for helping produce deep non-REM sleep. And the kind of sleep that these animals have is light, fragmented, short. They rouse very easily. And the clinical relevance of this is that with normal aging, and particularly in Alzheimer's disease, there's a loss of these preoptic neurons, and people's sleep becomes more fragmented and light. And so it's quite possible that this system is starting to fail as a function of age, and in some neurodegenerative diseases, contributing to the poor sleep that probably makes well, in the case of Alzheimer's, it may well contribute to more and more cognitive impairment in Alzheimer's. So we can take all these circuits and tie them up in a very simple little model here. And so on the right-hand side, we can just lump together the monoamine systems and the acetylcholine systems that promote wakefulness. And on the left is the ventrolateral preoptic area and the other sleep-promoting systems. And I mentioned to you how the ventrolateral preoptic area plays, its main role is to inhibit the wake-promoting systems. It uses GABA. It also uses a peptide called galanin. What I didn't tell you is that the monoamine systems and acetylcholine inhibit these sleep-promoting cells. And so that actually is a characteristic of all those sleep-active neurons, is they're inhibited by norepinephrine and other similar wake-promoting neurotransmitters. And the cool thing about this is that you end up with this really simple circuit that's mutually inhibitory. And what we think is going on is that during wakefulness, the monoamines are driving wakefulness, but they're also shutting down the sleep systems. And it really helps, we think, produce full wakefulness without some sort of muddy in-between state. And similarly, during sleep, once you transition into sleep, the sleep systems are inhibiting the arousal systems, helping keep you asleep. And so we think this helps produce these distinct states. You can think of it as sort of bistability, where you're either awake or you're asleep, and you're not spending a lot of time in between. And so this little circuit is great for producing the extremes, fully awake or fully asleep. But one of the problems with this is that how do you actually stay awake across the day? Because what if there's a little bit of homeostatic sleep drive that accumulates across the course of the afternoon? And, because if a few of those preoptic neurons start to turn on, they could then start to turn off some of the wake systems, and you could transition into sleep at an inappropriate time. And similarly, how is it that circadian drive is integrated into this? And so we're going to come back to this question of how do you stabilize this sleep switch so that you're fully awake across the day and then you stay asleep across the night. But before we get to that, I just want to talk a little bit about REM sleep. And REM sleep, of course, is pretty obvious. It's a state of cortical activation. People frequently dream in REM sleep, and the dreams are vivid and emotional, and they're paralyzed, right? When you're in REM sleep, your body is completely paralyzed except for the muscles of respiration and the eye movements, because there's little dashing eye movements that occur in REM sleep. And autonomic tone is quite labile, and so you can see heart rate going up and down, and same with blood pressure. The pathways that control REM sleep are predominantly in the pons. And it's probably easiest to tell this story starting off from the sublateral dorsal nucleus, which is in the mid-pons. And so that region you can kind of think of as ground zero for the paralysis systems. And how it works is it sends descending projections down to the ventromedial medulla to inhibitory neurons, which then inhibit motor neurons. And so when the sublateral dorsal nucleus turns on in REM sleep, it activates this inhibitory system, and it just completely suppresses activity in the motor neurons, so that even if an animal wants to move as a consequence of some activity in a dream, it can't. There's also, we're not entirely clear where those inhibitory neurons are. There's probably some in the medulla, there's probably some in the spinal cord that are premotor neurons, but they're both inhibitory, and they really hyperpolarize and inhibit the motor neurons. The reason the sublateral dorsal nucleus is so important is because if that area is lesioned in animals, animals will act out their dreams. And it's a pattern that looks an awful lot like REM sleep behavior disorder in humans, where the paralysis of REM sleep fails. Those sublateral dorsal neurons are probably turned on by a subpopulation of the cholinergic neurons in the pedunculopontine and lateral dorsal tegmental nuclei. And those guys, as I mentioned before, are important for activating the thalamus, because in REM sleep, you got to have a lot of cortical activity, just like during wakefulness, and you got to turn on the thalamus, and so the cholinergic system is important for that. So these two things together, the cholinergic neurons and the sublateral dorsal neurons, are important for ultimately driving the mentations and the paralysis of REM sleep. During wakefulness, these REM active systems, REM promoting systems, are inhibited by the monoamines. And so histamine, norepinephrine, and serotonin neurons all strongly suppress activity in those REM promoting systems. And this is important because when we give patients an SSRI, like fluoxetine or something like that, it's very common to see a very, very strong suppression of REM sleep. And we think that's occurring because you're having persistently high serotonin or norepinephrine that's shutting down the REM active systems. There's also another cell group that I haven't mentioned yet, sort of in the middle of the picture there, the ventrolateral periaqueductal gray. That's also another important REM inhibitory region, and this one uses GABA. And there's nothing that we have right now that really targets that, but we do think it's actually quite an important system for helping shut down REM sleep. So I mentioned before this puzzle of what stabilizes wakefulness across the day. And we think that really one of the most important factors contributing to this is another neurotransmitter system, which was discovered by two groups simultaneously. Tom Kilduff, Louise Delathea, and others named this Hypocretins. Yanagasawa and Sakurai's group named this Orexins, and the two names have stuck. And so, we use them interchangeably. This is a peptide neurotransmitter system, so it's not little small molecules like glutamate and GABA, but these are in fact, small proteins about 30 amino acids long, and they come from the same precursor protein. So one single transcript coming off of a messenger RNA makes orexin A and orexin B, also known as hypocretin 1 and 2. And then it binds to the two orexin receptors, OX1 and OX2. And the effects of the orexins on neurons are generally excitatory, and so we're just going to sort of lump it all together and just say orexins excite neurons. One of the things that's fun about this system is anatomically it's super tidy, and so you can see the orexin or hypocretin neurons in that cover of cell over on the right-hand side there. Orexins are basically only made in this cluster of neurons in the lateral hypothalamus, the white dots that you can see that sit sort of astride the third ventricle there. And on the bottom panel, you can see a sagittal view with the olfactory bulb at the left, medulla on the right, and you can see the white orexin-producing neurons in the middle there. And so these orexin neurons are just found in this one little cluster, but they innervate much of the brain, and they even go down into the spinal cord. And importantly, some of their most striking targets are a lot of these regions we've just talked about, particularly the wake-promoting cell groups. And so there's very strong orexin innervation of the histamine neurons in the tuberomammillary nucleus. The locus ceruleus, the raphe neurons get very strong innervation. And then that area I mentioned a couple slides back, that REM-inhibiting region, the ventrolateral periaqueductal gray, also gets strongly innervated by the orexin system. And so if you inject orexin into the lateral ventricle of a rat, that rat will stay awake for about 4 hours continuously. It's a really strong effect, and they'll have no REM sleep for about 6 hours. And so I think you can really sum up the effects of the orexins as they help sustain long periods of wakefulness, and they strongly suppress REM sleep. And the reason this is so important is because in narcolepsy, the orexin neurons die off. There's about a 90 to 95% loss of the orexin neurons in a very selective fashion, and the results have a very strong impact upon sleep architecture. And so up top you can see a hypnogram from a healthy control individual who falls asleep about midnight and has that typical pattern of non-REM and REM alternating across the night, with the REM shown in this sort of pink color. And then that person wakes up around 8 in the morning. Somebody with narcolepsy on no medications who was instructed to fall asleep at midnight is falling asleep before midnight and immediately going into REM sleep. And this is a very common feature of narcolepsy, these very fast transitions into REM sleep, because you can just sort of imagine the brakes are off on REM sleep. It can occur at any time. It's lacking the whole circadian control that's typical of REM sleep. And you can go have these very rapid transitions into it. Across the course of the night, this person with narcolepsy has very fragmented sleep with many awakenings. And even after they get up for the day at 8 in the morning, they continue to have naps during the day, and many of those naps contain REM sleep. And so the real features that pop out on this hypnogram on the bottom are poor maintenance of wakefulness, can't stay awake for long periods in the day, lots of REM sleep during the daytime, easy entries into REM sleep, and fragmented sleep at night. And it's probably all due to this loss of the stabilizing influence of the orexins on this circuitry. So we can add the orexin system to our little circuit diagram. And as some of you might know, I work a lot on the orexin system, and I think it's so important, and that's why it has this sort of, important position on the top of the circuit. Honestly, it's probably no more important than many of the other elements here, but it's something that we think about a lot, and I think it plays a very crucial stabilizing role, which is a little different than some of the other systems. And so the orexin neurons, as I said, heavily innervate and excite the other wake-promoting brain regions, and they're active during wakefulness, and so we think that they're important for helping sustain long periods of wake. It's a little controversial, but many labs also think that the orexin neurons are auto-excitatory. where they use orexin and glutamate to turn themselves on, which is really an unusual thing in the nervous system. Most neurotransmitter systems are autoinhibitory. The serotonin neurons do it through 5-HT1A, the dopamine through the D2 receptor. And, but if you record from orexin neurons in a brain slice and apply some orexin, the cells will fire faster. And so it really sort of raises the possibility, well, Tony Vandenpoel raised this idea some years ago, that maybe once orexins are released, they actually activate the orexin neurons, and that helps produce sort of a feed-forward system where they continue to fire, and that sustained activity helps drive sustained activity in their target regions, the other wake-promoting systems. It's also possible that the orexins help stabilize wakefulness simply because it's a peptide neurotransmitter, and peptides tend to stick around longer than some of the shorter-acting classic neurotransmitters, and that may also help produce sustained effects. They're also acting through G-protein coupled receptors, and so there's more long-lasting effects on the target neurons too. So this is a quick summary of the stuff that we've just talked about in the last half hour or so. And this is showing patterns of activity in the various cell groups, and I think it helps just sort of reinforce some of the ideas. The monoamine neurons tend to fire a lot in wakefulness, a little bit in non-REM, but they're silent in REM sleep. And that silencing of the monoamine systems is essential for allowing REM sleep to occur. The cholinergic neurons are basically wake active, but they also turn on to some degree in REM sleep to help produce the active mentations of REM sleep. Orexin neurons are easy, they're just wake active. And then you have these GABAergic systems in the preoptic area and elsewhere that are helping drive both non-REM and REM sleep. And so there's, what I've described here is this switching mechanism that helps drive states, but on a more macroscopic level, we have to think about, well, how do you get that total 24-hour pattern of sleep and wakefulness? And we certainly know there's a homeostatic component, right? The longer that you're awake, the sleepier you get. And so there's some research now on what are those underlying neurochemicals that drive that. Adenosine is one that's gotten a lot of attention. There's also a circadian component, right? If you stay up all night, you feel terribly tired at 3 in the morning, but if you continue to stay awake, 10 o'clock the next morning, you actually often feel better. And that's because there's circadian waking drive that kicks in. And so I don't have much time to focus on this, but I think you're all familiar with what circadian rhythms are. Here's just some examples of the rhythms in body temperature, growth hormone, and cortisol. And the important thing with circadian rhythms is they persist in the absence of any external light cues, right? And so you could put somebody in a dark room and these rhythms would persist because they're internally driven by the suprachiasmatic nucleus. And so the suprachiasmatic nucleus sits just above the optic chiasm. It receives signals related to external light coming in from the retina, which helps synchronize the SCN neuron activity to the daily light-dark cycle. And then these neurons send projections to lots of brain regions to help drive sleep-wake rhythms, temperature rhythms, and the rhythms of melatonin and other hormones. And so just a really quick thing here. The SCN is not directly connected to the sleep-wake circuitry, and you don't want it to be. Otherwise, your sleep system would be just locked into the daily light-dark cycle. It's relayed through a couple of other nuclei, the subparaventricular zone and the dorsomedial nucleus of the hypothalamus. And then those relay the signals out to the orexin neurons, the locus coeruleus, and other places. And this is important because having a couple more synapses in there gives the body some flexibility so that animals, for instance, can adjust to seasonal variations in day length, or you could switch activity to nighttime or daytime depending on the animal's behavioral needs. So I want to just wrap up on the last couple of slides by putting this in a sort of a pharmacologic context and thinking about the drugs that we have and the drugs that we might have in the near future and how they tie into these circuits. And so here's the same circuit diagram, and I just want to sort of plug in where some of the things are that we're using in clinic or hope to use soon. And so down at the bottom right there, you can see some of the drugs like amphetamines. They're predominantly acting to promote wakefulness by enhancing dopamine activity. Modafinil also increasing dopamine. Solriamfetol, which is under development by Jazz, is increasing both dopamine and norepinephrine. Skipping up to the top, orexin agonists, which are just now starting to go into clinical trials, are going to be enhancing orexin activity. We don't actually have any clue exactly where sodium oxybate works. It's been around for a long time now, but it's still quite a mystery of how it actually has its effects. I'm putting question marks here because I have this feeling, just based upon some very early data, that it may, it's a super sedating medicine we give at night, and there may be some rebound hyperactivity in some of these arousal systems during the daytime, but that's just pretty speculative at this point. For sleep-promoting drugs, for the last couple of years, we've had clinical use of orexin antagonists like suvorexant. Of course, the vast majority of drugs that are out there are, benzodiazepines and the Z drugs like zolpidem, zaloplan, and those are predominantly enhancing GABAergic signaling. Whether they really work on these preoptic pathways is anybody's guess, because there's GABA receptors all over the place. But it's possible that that's certainly one of their main effects. And I think the acute sedating effects of sodium oxybate could easily be through these pathways too. And then you can also promote sleep by blocking those wake-promoting neurotransmitters with antihistamines, anticholinergics, and drugs that block dopamine signaling. So just to wrap up the main messages here, wake-promoting drugs are mainly working by increasing monoamine tone, dopamine tone, norepinephrine, histamine, and so forth. Though they're not yet clinically available, we're hoping in the next few years to see drugs that promote orexin signaling. And conversely, the sleep-promoting drugs mainly enhance GABA-A signaling. Sodium oxybate's the exception of that. It's probably more of a GABA-B drug. Or they block monoamine or orexin signaling. And I didn't have time to talk about it, but of course melatonin is also used very commonly to help promote sleep, though I think it's probably best thought of as a chronobiotic, as its direct sedating effects are not particularly strong. So that's it for my part. And I think, Andy, if we have time, I could take some questions now, if people want. Sorry for the blizzard of acronyms and neurotransmitters. It's a tough thing to swallow first thing in the morning. Any questions? Yeah, Tom.
You talked about the various arousal signals and sources of emotion. The top one was attentional. I'm not sure what you meant by attentional arousal.
Yeah, I think so. I guess the thing is that when, as sleep researchers, we look at the EEG and we say that's wakefulness, but there's 1,000 different types of wakefulness. And so I think it's important to think about what aspects of wake you're promoting. Yes, you may be increasing wakefulness, but what's the brain doing along with that, right? Is it, for instance, if you could give a drug that really cranks up noradrenergic tone, and you'll probably get great attention, but you'll probably get a lot of anxiety with it too.
So when you say like serotonin is motivational arousal, right? That was the second. What data?
Yeah.
So are you saying that it increases emotional response or increases wakefulness through emotion?
Yeah, I don't really know. I mean, honestly, I'm speaking of it in just very loose terms. I really think this is actually this is something that we need to work on the basic science level and clinical as well, because I think it's something that as sleep researchers we haven't really talked about a whole lot.
I agree. I think it's a very useful taxonomy.
Yeah, no, I mean, certainly, like for instance, when we give patients amphetamines, they say, I'm awake, but I feel like my spring's wound up, and it's not normal, comfortable wakefulness. It's an unusual, artificial kind of wakefulness. Yeah. Atul?
Can you talk a little bit about sort of synergy between drugs? Because for either sedatives or stimulants, I think some combinations of drugs seem to work better in the clinic. And maybe to the last question from Tom, maybe there are different aspects of wakefulness that can be promoted with different drugs. Are there combinations that you think are rational?
Were you thinking of something in particular?
No, I just think clinically, like for example, sodium oxybate plus modafinil seems to work really well clinically, and I'm not entirely sure why. It just seems an observation. But given that there are different kinds of wakefulness, maybe you could promote them with different agents. Yeah.
Well, and certainly I think to take that example in particular, modafinil is essentially blocking dopamine reuptake, and I don't think that has anything to do with sodium oxybate. And so, I guess on the plus side, one of the reasons why that combination seems to work pretty well is because you're using completely independent mechanisms to ultimately promote wakefulness. And I agree with you, we can often get pretty good traction on helping patients feel better with that. And I think it's just simply because you're engaging more than one system. I think, one of the great challenges, for instance, with just plain old amphetamines is you're essentially relying upon one system to do all the heavy lifting to try to generate full wakefulness when the brain is desperately trying to fall asleep.
should we be trying to promote different aspects of wakefulness to get fully awake state?
Yeah, well, certainly in a pharmacologic sense, you're driving it in different ways, and whether the kind of the flavor of wakefulness is truly different with that, I don't know. One of the this is completely anecdotal, but sometimes like with sodium oxybate, patients say that the wakefulness feels very different than amphetamine wakefulness, and what that is truly is really an interesting question.
Is that also true for the sleep side? You sort of talked about, like, with a histamine antagonist and a serotonin antagonist, you very much.
Yeah, no, I think that's a great question. And again, it's one of these things that, in the same way that we haven't really distinguished between the wake drugs, we haven't really distinguished a lot between the sleep drugs either. And so how does benzodiazepine sleep feel different from Ramelteon sleep?
Well, the drugs you talk about, the psychiatric drugs, the antipsychotics, drugs like amitriptyline, which hit multiple of these receptors, it's an antihistamine, it's an antiserotonin, so it works on lots of different systems.
Right. So, well, I guess, certainly, again, speaking more from clinical experience than from the basic science, a lot of patients really like benzodiazepines because they can produce a certain degree of oblivion. And, one of the reasons that, for instance, like Ramelteon has not been such a hit is because while it may be somewhat sedating, it doesn't give people that deep unconsciousness that you can achieve with benzodiazepines. And so it's, obviously we don't want that, right? We don't want people completely stuporous. But if somebody with insomnia has a tendency towards waking up frequently during the night, that's sometimes hard to get around if you're using a gentler drug. And I say the same thing for suvorexant too, where people tend to wake up. And Tom and I had a paper this last year where people on suvorexant still wake up just as often as on placebo. They just don't stay awake as long. And so in this case, it's an orexin antagonist. Orexins help produce long periods of wakefulness. And so when you block the orexin system, it's great for getting back to sleep. You don't see long awakenings. On the other hand, the question is, how is that going to feel subjectively to the patient? Are they going to say, well, I'm still waking up just as much? Or will they appreciate the fact that they're falling asleep quicker? And so I think it's just important to think about the distinctions between these drugs.